Hello, good afternoon, everyone. Let's get started. Today, we're going to be doing lecture 6, which is cellular reproduction. This is the first lecture of the second half of the course. In case you did not know, the course is separated into two parts, right? So you already did the test last week for the first half of the course, and those are going to be history. You will not be tested on lecture 1 to 5 anymore. For the remaining of the course, right? There's no final exam that tests you on everything in the course. There's only going to be one more test, which is test number 2. And test number 2 will only cover material from lecture 6 all the way to number 10. Right? So in a way, you can forget about everything that we did for the first half. You will no longer be assessed on those. Okay. So today we will be talking about cellular reproduction. So let's start with a cell. Okay, in the cell theory that we learned before, one part of the cell theory states that all cells come from the division of a pre-existing cell. Okay, so let's say we have one cell here. We call this the parent cell. Parent cell. All right, and let's just say, for simplicity's sake, the parent cell has 4 chromosomes. So 1, 2. Okay, so one pair of chromosome 1, and then it will have one pair of chromosome 2. Okay, so this is a parent cell, a total of 4 chromosomes. Okay, 4 chromosomes. And what happens is right before cellular division, right before mitosis, we have to do DNA replication. So during DNA replication, all the genetic materials are going to be copied. And afterwards, right, we're going to have this much genetic material. Okay, the chromosome that has been copied are going to be stuck together, right, at the centromere. And there we go. That's another one, and that's another one. Okay, so there is some confusion here that is very common. Even I get confused sometimes. But how many chromosomes do we have here? Okay, we started with 4 chromosomes. So after DNA replication, how many chromosomes do we have in this picture? Okay, Cedar says 8. Anyone else? Moira said 4. Alex said 4. Anyone else? Let's do a vote. Okay, because we always do polls. Right? If you think it's 8, please choose A. If you think it's 4, please choose B. If you think it's other, please choose C, and then type your answer in the box, in the chat box. Okay, we are neck to neck right now. Half people think, half of you think is 8, half of you think is 4, and I'm waiting for 6 more responses. If you are here, please choose something. Is it 8? Is it 4? Is it something else? Last call. Anyone else? All right. Well, the correct answer is 4. Okay, 4 chromosomes. And this might sound strange, okay? But the people who come up with this concept, they say, as long as they're stuck together, that count as 1. Count as 1. Okay. So each of these is called a chromatid, right? TID. And so you would have 8 chromatid. I can write down for you 8 chromatid. But we will have 4 chromosomes still. Nevertheless, the amount of genetic material has been doubled after DNA replication. Okay, and so if we proceed to mitosis, we will have two daughter cells, okay, two genetically identical daughter cells. Okay, these chromatids will separate and it's gonna look like this. Do you all understand what I'm trying to say? Any question? Any questions? Do you need me to clarify? All right. If you think of anything, please feel free to unmute anytime. Now we're going to look at some key contributors in history that helps. They did some experiment, they did some discovery that relates to DNA. First, we have Chargaff. Right? I don't know his first name. Maybe you can Google it and find out later. But he has a rule, right? It's called the Chargaff's rule, and basically he's discovered that in any DNA molecule, the number of adenine will always equal to the number of thymine. And then the number of guanine will always equal to the number of cytosine. Now we know this already, right? This is called the base pairing rule, right? Okay. And the reason why they're equal is A always pairs with T and G always pairs with C, right? So given that concept, we know that the two strands of DNA, they are complementary. Okay, complementary. So that's what he discovered. That's Chargaff. Next, we have Rosalind Franklin. What she did was used X-ray to study the structure of DNA. Her data suggests DNA is a double helix. Unfortunately for her, she passed away before she can finish her work because of ovarian cancer, potentially because of the exposure to X-ray. They did not have as much protection back then compared to nowadays. So, she passed away before she can finish her work. And that's where Watson and Crick came in. Okay, they used Franklin's data to build a scale double helix DNA model. Okay, and if you search up Watson and Crick on Google Image, you will almost always arrive at the same picture. Right? It's them looking at the 3D structure that they built. Okay, Hershey and Chase. Anybody remember what Hershey and Chase did? Help me out here while I blow my nose. What did Hershey and Chase do? No? Okay, I'll tell you what Hershey and Chase did. They study, or we should say, they use bacteriophage. Which are these things that looks like that? They actually do look like that. Okay, this is a Bacteriophage, which is a virus that infects bacteria. Okay, so they use bacteriophage to show that DNA and not protein is the hereditary material. Okay. Lastly, we have Messelson and Stahl. What did they do? They used E. coli to show that DNA replication follows the semi-conservative model. Okay. For those of you who need a refresher on what semi-conservative means, right, so here we have the original double helix. That's the parent DNA. And after replication in the semi-conservative models, the daughter DNA will consist of half of the original strand and then another half that's completely new. Okay, so this is new DNA strand. And this would be the original DNA strand. Okay, so there were 3 models, right? If you saw the lecture, there's the semi-conservative model, there's the conservative model, and then there's the dispersive model. So their experiments show that it is the semi-conservative model that is the correct one. Okay. Next, we have write the complementary strand for the DNA strand below. Be sure to include directionality. And so here we have a strand that goes from 3' to 5'. The 3 and the 5 is just a way to indicate the orientation of the strand, the direction of the strand. And so on the other strand, we are going to start with 5, okay? Because DNA is anti-parallel, right? I'll explain what that means in a second if you're confused. And then we're going to use the base pairing rule. A pairs with T, T pairs with A, C pairs with G, and so on and so forth. Okay, and then we're going to end with 3'. Right? So this here, just to remind you, DNA is, or I should say that the two strands of DNA, they are anti-parallel. Okay, and what that means is they run the strands run in opposite direction. All right. Now let's try to illustrate this. Let's imagine you have a bunch of people who is lying on the floor, head to toe. Okay. Like that. All right. So here we will say this is head, this is toe. Okay, head to toe, head to toe, head to toe. And then lying next to each of these red people, we're going to have some blue people, but it's going to be upside down now. Okay, they're going to be holding hands. Okay, this one is going to be toe to head. Right, so you see, they're parallel to each other, meaning the distance between them is the same, but then the direction is opposite, right? From one side, we have head to toe. From the other side, we have toe to head. But in a DNA strand, you know, there's no head, there is no toe, right? So instead of saying head to toe, we say 3 to 5. And then on the other one is 5 to 3. Okay, exactly what the 5 and the 3 means, don't worry about it. We don't have to know it for this course. Okay, but it's anti-parallel. One strand is upside down relative to the other. Okay. All right, let's take a look at the next question. If 23% of all a cell's nucleotides are adenine, what would be the percentage of the rest of the nucleotide? Well, we did something similar like this before, right? But let's just go through it again. If you have 23% adenine, that means you're going to have 23% thymine, right? Because Chekhov's rule says that percentage of A, number of A's equals the number of T's, right? And if you add them up together, if you add these two together, the A and the T's, you're going to get a 46% of A's and T. So in total, we're going to have 100%. And if you take 100 minus 46, you're going to get 52, right? Sorry, 54. 54% is C and G. So if you divide this by 2, you would get 27% being C and 27% being G. Okay. Are we okay with this concept? All right. 4 stages of the cell cycle. We have G1, we have S, we have G2, and we have M. So over here, the first 3 stages, and that is what we call interphase. Right? So that's just when the cell is like growing, performing its function, so on and so forth. Okay? G1 and G2 are like checkpoints, which we'll explain in a second. S is where you will do the DNA replication. And if all goes well, then we're going to go on to M, okay? And M is mitosis, right? So the cell spends most of its time in interphase, right? Most cells spends 90% of its time in interphase, and then it's going to divide when it has to. All right. So let's take a look at the G1 and G2. It's like getting your driver's license. You got to do your G1. You got to pass your G1 first before you can do your G2. Okay, but then there's going to be a little bit of wait time in between. So let's take a look at what's going on in G1. Okay, so in checkpoint number 1, G1, we want to check for a couple things. For example, we want to check for like cell size. Cell size. Like, is the cell big enough to divide? Okay. One of the main reasons of why a cell needs to divide is when it gets too big, it's no longer efficient in transporting stuff within the cell. If it's really big, you don't have enough, membrane surface area to help maintain the efficiency of the cell. And so you need to split it apart. Okay. You don't want to split the cell. You don't want to divide the cell too early because you might not have enough cytoplasm. Okay, so one of the things that G1 checks is to see if the cell is big enough to divide. Okay, another thing that it checks is nutrients, for example. Right? Are there enough resources, right? And perhaps ATP for mitosis. Okay, it's a very energy demanding process. You got to move those chromosomes around. You got to copy the DNA. Do we have enough nucleotides, right? Do we have enough ATP to drive this process? Right? If the cell is poorly nourished, maybe it's not a good time to divide. Okay. What else are we going to be checking in G1? Anybody want to help me out here? What else should we check in G1 before we proceed to DNA replication? No? All right. We want to check for presence and perhaps absence of growth factors or inhibitors. Okay, for various reasons, there are all these stimulus that are going on in the cell, right? Some stimulus are going to trigger the cell to divide. Others are going to suppress the cell from dividing. Okay, and it comes in the form of what we call growth factors or growth inhibitors. Okay, so you want to check for the balance of these things. Okay, maybe there is a lot of inhibition going on at this moment, then we're not going to divide. Okay, maybe there's a lot of growth factors that promotes division, then we want to go ahead and do it. Okay, so that's one of the things it checks for. And finally, we also want to check for DNA integrity. Okay, are there any damages? Right, we want to check for things like telomeres length. If you don't know what telomeres are, I'll explain later. Okay, so all these things. Right? So if everything is good, we're going to go ahead and proceed to the S phase, where you will be copying all your DNA. Okay. And afterwards, we're going to proceed to another checkpoint, G2. So in G2, we want to check for, again, DNA integrity. Right. When we copy the DNA, we got to unzip it. Right? We got to put it back together. We just have to make sure that no damage was done to it during the copying process. So again, any damage. Okay. We also want to check for DNA errors. Okay. Was there any mistake? During the copying process. Okay, we are talking about copying like millions and millions of nucleotide. Right? It's not uncommon to make a few mistakes along the way, right? And then that's okay. You sometimes can correct them, and sometimes you don't. If you don't correct them, you end up having a mutation, right? Okay, but you check for these things in G2. So if all goes well, then the cell is allowed to go into mitosis. Now I should make a note here. If the cell fails G1 or G2, okay, you won't fail both, right? Because if you fail G1, you automatically will not proceed to S. Okay, so you might pass G1 but not G2, okay, or you will not pass G1 and then the process will stop right there. Okay, so if the cell fails G1 or G2, then it will trigger apoptosis, which is programmed cell death, or growth arrest. Okay, so it either go kill itself or it is going to enter growth arrest, which means it is not allowed to divide anymore. Okay. Any question? I should probably tell you that this is also called senescence. Okay, it's one of the reasons why we age. When we get older and older, the cells, they just don't divide anymore, and then they enter senescence day. And if we have a large pool of cells that is in a senescence day, then we age, sadly. There's a question. What happens in S phase? In S phase, that is where the DNA replication occurs. DNA replication. Okay, with the whole helicase and gyrase and all the stuff that we learned in the lecture. We good? All right. List the 4 stages of mitosis. What's going on in mitosis? Well, we're just going to list them right now, and we can talk about what happens in them later. We have the prophase, followed by metaphase, and then anaphase and telophase. Okay, so the order is PMET. If you need to have a way to remember that. I think it's pretty straightforward though. You should be able to memorize it without an acronym. Prophase, metaphase, anaphase, and telophase. Okay, so we'll come back to talk about what happens in each phase later. But right now, we will talk about all the enzymes and proteins that are necessary in the process of DNA replication. Okay, so this is DNA replication in eukaryotes, right? Okay, so like a human cell, for example. All right, so first we have gyrase. Okay, gyrase. What does gyrase do? Just gonna find a finer tip here. Okay, gyrase. The function of gyrase is to relieve tension that builds up in the DNA as it is being unwound by helicase. Okay, so when you unzip the DNA, it is going to create some tension in the strand. And if we do not relieve those tensions, then we risk snapping the DNA molecule, breaking it. Okay. So gyrase will make little cuts here and there in the sugar phosphate backbone, and it will relieve the tension and prevents DNA breakage. Prevent DNA from breaking. Okay. Helicase. We switch to a different color. Helicase is the enzyme that unwinds the DNA to create a replication bubble. Okay, replication bubble. Helicase move into the replication fork as DNA is being copied. Now, after you pull the DNA strands apart to create a bubble, the DNA strands actually have a tendency to come back together because the nucleotides are attracted to each other, right? We have the A, C, T, G on one side, and then on the other side, we might have like T, A, G, C. And those nucleotides are attracted to each other because of the hydrogen bonds that they're going to form. We don't want them to come back together though, okay? Because we need them to stay separate in order for us to copy the DNA, right? So that's why we have these single strand binding proteins, and the function is to bind to the sugar phosphate backbone of the separated DNA strand. Okay, and the job is to prevent reannealing. That's what it's called. Okay, prevents them from coming back together. So I'll give you a moment to catch up copying all these things, and then we're gonna move on to another diagram in a second. Okay. All right, so this diagram, right, at the very end, okay, let's take a look at here. What number represents the helicase? Can someone tell me what number here represents the helicase? Number 2? Yeah, that's right. Okay, right here, number 2. That is the helicase right here. Okay. Okay, so the helicase is gonna create a replication bubble, and then this location here is called the replication fork, right? And then it will keep on moving along this way to to open up the DNA more. Think of the helicase as like the I don't know what you call that thing, but the thing in the zipper that you hold on to unzip your jacket. That's essentially what this is. Okay, you keep on pulling it down, and you will continue to unzip your jacket. What would number 1 be then? This little guy here. What is this thing? Yes, Ueda said is gyrase. That is correct. Okay, and the function of a gyrase is to clear up any knots that is going to be building up as the helicase unzip. Okay, so we don't want any tangles, any knots that might break the DNA. And once you separate the strands, we got to have these little orange balls. Okay, the orange balls, they are the single strand binding protein. Right? They keep the strands apart so they do not come back together. They prevent it from reannealing. Okay. All right. Let's go back and write more, and then we'll come back to this diagram. I hope you guys are doing okay. I mean, I can't really tell. Usually in the classroom, I can just look at people's face and I know if they're with me or not. But unfortunately, I cannot do that right now. Okay, I'm going to skip the primates for now, and I'm going to talk about DNA polymerase 3. Okay, so what is DNA polymerase 3? This is the enzyme that will use the parent strand, use the parent DNA as a template to create a complementary strand. Okay, so it uses the original strand, and using base pairing rule, it will know how to create a new strand. Okay, but there are 2 limitations here for our DNA polymerase. Okay, I'm going to write them down for you. 2 limitations. Anybody remember what the limitations are from the lecture? Help me out here. Please, please. What is the problem with this thing? 2 problems.
It needs something to help it get started.
Exactly. Thank you, Moira. It doesn't know where to start. Okay, so cannot initiate the process, the replication process. It doesn't know where to start. It's like my daughter came home, does not know where to start for her homework. I have to open up her workbook, tell her, do this page. And then she would start there and carry on by herself. So something needs to tell DNA polymerase 3 where to start copying. Okay, that will be the job of the primates, which we'll talk about in a second. There's a second limitation, and I think Adriana said in the chat, DNA can only build in the 5' to 3' direction. Exactly. Okay, so DNA polymerase can only build in 5' to 3' direction on the new strand I'm talking about. Okay, the new strand that's being built has to be built in the 5' to 3' direction. Okay, so that creates a little bit of a problem. All right, before we go to the diagram at the end, let's just quickly talk about primates a little bit more. Okay, so primase is like, it's like the parent that tells the kid where to do the homework or where to start the homework, right? So primase will create a short RNA primer, right? To help position the DNA polymerase 3. So that it can start the copying process. All right. The replication process, whatever you want to call it. The replication sounds better. Okay, let's go back to the diagram here. I think that this is probably the primase. Okay, it's kind of hard to tell, but believe me when I tell you, this is the primase. Okay, so what is the primase going to do? The numbers are not really referring to the structure per se, right? The numbers is actually for you to write down what's happening. Okay, so like you're supposed to write down 1, gyrase will remove any tension, blah, blah, blah. And then step 2, step 3, so on and so forth. So like, you know, that's why there is number 5 pointing here and number 5 pointing here, right? Don't get confused by those. Those are just like events that are happening. Okay, so the primase is it's gonna lay down the RNA primer, right? So there's gonna be an RNA primer here. It's gonna be another RNA primer here and another RNA primer here probably. Okay, and then one RNA primer here. Okay, the primase are not shown at all locations. Okay, so let's take a look at here. Do they have a DNA polymerase 3? I think this one is the DNA polymerase 3. Okay, so this thing here, this blue guy. Let me try that again. This is the DNA polymerase 3. Oh my goodness. Polymerase 3. Okay, and you can see it is building the new DNA strand. Okay, let me use pink for the new DNA strand. It's going to go this way. Now pay attention here because it can get a little bit confusing. Okay, and it's actually extra confusing because they decided to create this twist here. I think the twist is meant to show you that the gyrase is going to relieve that tension. Okay, but let's chase this strand. Okay, this is 5 prime. If you chase it all the way, right? Okay. Oh, actually, did they made a mistake here? You know what? Let's just ignore that part here. I think this might be a little bit confusing. But you can see at the bottom here, right? This is 5 prime all the way to 3 prime. So which means when I build a new one, when the DNA polymerase is going to build a new strand, this is the 5 prime end, and then this is the 3 prime end, right? Oh, sorry. I think there is something wrong with the diagram here. Let me just take a moment and make sure. Yep. Okay, this is the 5 prime end, and then this is the. Oh, hold on. 5 to 3. Oh, okay. I understand why it's confusing here. Okay, so the primase will put the primer here. Okay, and this is going to be 5 prime end to 3 prime end like this. Okay, but as you open up the fork more and more, right, you will have to put another primer here, and then now you're going to have another 5 prime to 3 prime, and then you will open up more, and you will have another 5 prime to 3 prime. Okay, so like when I open up even more here, I will need to have another primer, like over here before I can copy, right? So what that means is this strand at the bottom, it is not being built continuously. Okay, so like you have to build them in a little bit at a time. And so we call this the lagging strand. Yeah, the twist was confusing me, sorry. Maybe I will take this away in the future. So this here is the lagging strand. Lagging strand, which means it is not being copied, continuously. Okay, rather it is being copied in fragments. Okay, so you probably know by now that these fragments, they're called Okazaki, right? Okazaki fragment. Okay. Whereas on the other strand, right, it's going from 5' all the way to 3', and it's no problem. You just need to put down one primer, and it's just going to keep on going and going and going forever. Okay, so this is called the leading strand. Leading strand. Okay, and this is being copied continuously. Okay. So sorry about the little confusion at the beginning. Hopefully I did not, confuse you too much. But now it's time for question if you need clarification. Before I move on to the next part, does anybody want to ask me to clarify anything? Or are we still together? Yeah, there is an animation, right, that online that I post. So probably it will be a good to watch that. There's a question. Yeah, primates, gyrates, anything that ends with ASD is enzyme. Basically, all these are enzymes except for the single strand binding protein. Okay. All right, now here's the next part, right? And again, it's talked about in the lecture, but I'll repeat it. Okay? The purple stuff here, this purple stuff here, that's a RNA primer, right? Okay, it's made up of RNA, right? So for example, it could be like AUCG or whatever, right? There's uracil in it. So there is a RNA primer here, RNA primer here. Okay. So what happens is this. The DNA polymerase 3 will continue to build the strand. Right? It will start where the primer is, right? Because the primase kind of prompts it to do that. And then it will continue to build. As it's building the strand, eventually it is going to reach the primer that was laid down by another primase. Okay, so we need to replace these RNA primer. We cannot have a uracil in our DNA, right? So as the DNA polymerase 3 approaches an RNA primer, DNA polymerase 3 will fall off. Okay, and after it falls off, another polymerase will come on, and it will take off all these RNA primer, all these purple stuff, and replace it with actual DNA nucleotides. Okay, so that enzyme is not shown here, but if you saw the recordings, you would know I'm talking about the DNA polymerase 1. Okay, so over here, DNA polymerase 1, right? It will replace DNA polymerase 3 when DNA polymerase 3 approaches an RNA primer. Okay. And then the function of DNA polymerase 1 will remove the RNA primer, right, the purple stuff, and replace them with DNA. We are almost there. Finally, all those Okazaki fragments needs to be connected together, right? And that is the function of ligase. Okay, ligase will join the Okazaki fragments together. Okay. And seal all the gaps in the lagging strand. Okay. Now, I'm going to talk a little bit about telomerase, but in order to understand that, we have to draw a picture first. All right. So hopefully you got everything, and if you didn't, that's okay. I'll post it online afterwards, anyways. Okay. Now you don't have to draw this. You can just, watch and ask questions if you have any. Okay, so this is my DNA. Right? This is original DNA, double strand. And what's going to happen is we're going to open it up into a replication bubble, right? Okay, with a helicase and all. All right. So let's say this is 5 to 3. This is 3 to 5. Okay. No weird twist this time. Should be very easy to understand. And so, right, what we talked about so far is we got to put a primer here, right? Okay. So we're going to put a primer here. And the primer is going to be, of course, anti-parallel. So if this is 5 to 3, right, then this time this is 5, and then this direction is 3, right? So the polymerase 3 will come, and it will keep on building into the fork like that, right? 3', no problem. Okay. And then on the other side, right, at the beginning, you will put a primer here, right? This is 5 to 3, and this side will grow into the fork. But as you open up more and more, right, you're going to have to put another primer and another primer, and then you will have this and this. So basically what we talked about, right? Okay, same thing over here. Right? You're going to have to put some primer, and then over here. Right? So this actually brings out another good point, right? You can see the replication bubble on the left side. The top is the leading strand, isn't it? This is leading strand. And then the bottom is the lagging. Do you guys know how to tell the difference? Leading and lagging? Yeah. But if you look at the other side of the fork, right? Now that becomes the lagging. And then this is the leading one, right? Okay. Now let's take a look at what happens. Okay. So suppose I open the fork all the way to the end, right? So I am going to need to have a primer here, right? And then DNA polymerase 3 will come, and then it will replicate this section, right? And then if I keep on opening the fork, then this is just going to keep on going all the way until the end, right? So here is a problem. Here's a bit of a problem. Okay, let's take a look at the lagging strand at the bottom. The DNA polymerase 3, which I did not draw, will fall off when it approaches the primer, and then polymerase 1 will replace these purple primers with actual DNA. So this purple part will become DNA later. This purple part will become DNA because, again, right, the polymerase is coming down, right? This purple primer will also become DNA. However, we have a problem at the very end. There is no polymerase coming from upstream, we say, okay? So this primer here, this little bit of primer, this primer will not be replaced by DNA, and it just kind of falls off and not get replicated. Does that make sense? Why this part would not get replicated? Right? Because there's no DNA polymerase 3 coming from above, right? From upstream. So this part will not get replicated. So what that means is every time you divide your cell, every time you copy your DNA, the chromosome gets shorter and shorter and shorter. Okay, so if you look at the chromosome here, right, after each division, it will get progressively shorter and shorter and shorter. Right, so we cannot allow this. If it gets shorter and shorter, we're going to lose critical genetic information. So to prevent that from happening, to prevent the loss of crucial genetic information, you're going to have special non-coding DNA at the end, right? Those are called telomeres. Okay, so instead of the actual genetic material getting truncated, you are just going to be losing telomeres every time. So telomeres are nothing more than repeats of TTA GGG. It's just TTA GGG, TTA GGG, TTA GGG, like for like a couple hundred times. Okay, so every time you divide, you will lose a little bit of telomeres, until the telomeres get critically short. Okay, then the cell will not pass G1 anymore. Remember over here, right? We wrote down the checkpoint, right? DNA integrity. Any damage or the telomere length too short? If it's too short, you will not be allowed to divide, because if you divide another round, right, you don't have these buffers anymore, right? You're not going to be cutting off telomeres anymore. It's too short. If you divide one more time, you're going to start losing critical genetic information, right? So we don't, our body does not allow that, and the cell will enter senescence. Okay, will enter growth arrest. Is that okay? I hope it's okay. So what's the deal? Why are we talking about this? Well, in some cells, like germ cells, for example, germ cells are cells that would become sperm or egg in the body, as well as stem cells. Okay, in the germ cell and the stem cell, there is an enzyme called telomerase. Okay, and the function of the telomerase is to relengthen. Is that even a word? Relengthens the telomerase, the telomeres, by adding TTA-GGG to it. Okay, and it is expressed. That means it's only going to be found in germ cells. Germ cells, again, these are cells that will become sperm in male and eggs in female. Okay, some people like hear the word germ cell, they think it's talking about like bacteria and stuff. No, germ cells are cells that will become gametes, sperm or egg, expressed in germ cells and stem cells. Okay, so listen to me for a second, right? So when we are born as a baby, we have full telomere length. Okay, and then we age, we grow up. Okay, so my cells have been dividing for, I don't know, God knows how many times. It's every time it divides, it gets shorter and shorter and shorter and shorter and shorter. Okay, so like by the time I reach like mid-20s or something, my telomeres are going to be shorter than when they were when I was born, right? And this goes the same for everybody. And what happens is, if you decided to have a child in your late 20s or something, right, you cannot pass on your genetic material to your kids with shortened telomeres, okay? Because then they would be born with shortened telomeres, and then it will get shorter and shorter and shorter in their lifetime. And then when they have baby, right, the next generation, it will even be shorter, and then shorter, shorter, shorter. Eventually, we run out of genetic material, right? Basically, so that doesn't happen because of telomerase. So when we make sperm or egg in the germ cells, right, the short telomeres that we have at that point of life, they will relengthen. Okay, so that when you pass it on to the next generation, it gets reset in a sense, right? So they will start brand new. Do you guys know about Dolly? Dolly, the sheep. Dolly, the sheep. Anybody know what that is? Anybody heard of that? Who's Dolly? What's Dolly? They made a copy of her. Yeah, I think Dolly is the copy. I don't know if the mother is also called Dolly. Okay, but it's the first cloned sheep. Okay, so Dolly did not live very long, okay? And the way that they cloned Dolly was that they took a cell from the mother, okay? And then they shoved that cell into like the egg, okay? So they removed the nucleus of the egg, and then they put that, you know, a donor cell into the egg, and then they basically clone the sheep that way, okay? So because the cell that was taken from the mother is not a germ cell, right? It's just like a bodily cell. That cell has already lived for, I don't know, like let's say 8-10 years or something, okay? And 8 to 10 years in a sheep is comparable to like 20-30 years in humans, right? Okay, so that cell that was taken to clone the sheep has already been aged, so to speak, okay? So when Dolly was born, the clone sheep was born, it was not born with brand new long telomeres. Rather, it was the shortened telomeres that was inherited from that original donor cell. So what happens is, even though the sheep is young, it was born like, you know, today, but on a genetic level, on a cellular level, it is already old. That's why it didn't live very long, right? Because the cell would not be able to divide as many times as it would have been if it was like a brand new naturally born baby sheep. So it basically died of old age very, very quickly. Does that make sense? Okay, yeah. I don't know. I assume you guys have no question, then it's fine. So usually people are like, okay, so does that mean if we have a telomerase in the body, then, you know, we can live forever because the cells can keep on dividing and dividing, right? And, you know, we will never enter a senescent state, and, you know, we will live forever, right? Or at least extend our lifespan. Actually, that is not a very good idea, because in cancer, which is uncontrolled cell division, they actually have high levels of telomerase. Okay? Did you know if you take a cancer cell and you put them in a petri dish and you keep on feeding them nutrients, you can keep on dividing them forever and ever. Okay, they virtually are immortal. They will never die as long as you feed them. They will keep on dividing and dividing and dividing. In fact, you can buy these cell lines. They're called cancer cell line, and researchers, they can buy them online, you know, from like vendors, right? Not just on eBay, obviously. So they can buy them from vendors, and then that's how they study, you know, cancer treatments, for example. And all these cancerous cells, they have elevated telomerase, okay, which allows them to keep on dividing without truncating the telomeres, okay, and so they have unlimited replicative potential. So that's what I studied when I was in grad school. To look at telomerase expression in cancer, right? So one of the strategies people are trying to do is maybe they can come up with a telomerase inhibitor, right? And by blocking it, maybe it's going to be able to slow down the growth of tumors. Alex's question: with sharks, what makes their DNA so resistant to these type of diseases? Yeah, I've read that some animals don't get cancer, right? And you know, maybe sharks is one of them. I think elephant is another one. You know, they live very, very long, and then, you know, that tend not to get cancer. And obviously that's something that people are trying to find out, right? By understanding, you know, is there some kind of underlying cellular mechanism that makes them more resistant? If so, can we harness that and then, you know, maybe make that beneficial in the human clinical setting? So I don't actually know what the answer to that is, but certainly it's something that people are looking into. Yep. So let's take a quick break, and when we return, we're gonna do mitosis afterwards. Okay, so see you in 5 minutes. All right, and we are back for more biology. Now we're going to talk about the second half of the cell cycle business. We're going to talk about the mitosis part. Okay, so here we have number 8. Okay, identify and briefly explain the function of each cell structure shown in the diagram on the right. Okay, we'll do that. Let's choose a exciting color. Okay, right here. A. Okay, so A is pointing at the chromosomes. And you know, briefly explain. I guess chromosomes, they are the genetic code, right? They contain genetic code. Okay. B is pointing at these black circles. Those are the centromeres. Centromere. It is where duplicated DNA stick together. C is centriole. Okay. We learned this in what was it, lecture 3? Okay, so they produce spindle fibers. D is cell membrane. Not sure why that is here. Not going to describe it. We had a whole lecture on cell membrane. You should be well versed in what it does. And finally, E is pointing at the spindle fibers. Spindle fibers. Because, and what it does is it will guide chromosome movements. Okay, during mitosis. Okay, so we should quickly talk about DNA a little bit. Okay, so we know DNA in this form. This is the form that we are familiar with. Right? So that's the double helix DNA, okay? with nothing else attached to it. Now this is very, very long, the DNA. Okay, it will not fit in the nucleus. So what you want to do is you are going to package it. So you're going to add some packaging protein. Packaging proteins. Okay, and one of the most common packaging protein is called histone. Histone proteins. Histone proteins are positively charged, which is perfect for the DNA because the sugar phosphate backbone is negatively charged. So the together will stick very nicely together. So if you add the histone and the DNA together, then you're going to start to package the DNA into what we call beat on a string model. Okay, so the DNA will pack nicely like that. Okay, so this we call chromatin. It is DNA with packaging protein. And then during DNA replication, during mitosis, we are going to condense even more. Okay, condense more with more packaging protein. And ultimately, it's going to condense together into what we are familiar with. Okay, let's try to draw this thing. Okay, it's very, 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 very condensed. Okay, then we get our chromosome. Okay. So basically, it's all DNA and proteins, but depending on the level of how compact it is, we call them by different things. Okay, so these are, of course, well, the whole thing is a chromosome, but we know like this copy and this copy are identical, and we call these the sister chromatids. All right. Why don't we do some poll questions? We haven't done any today, I feel like. Let's try the first one. Let me go ahead. This one might not be too obvious yet because I didn't really explicitly state it, but you know, maybe you can do it. All right. Half of you have answered. I'm gonna end the poll here. Okay, so this whole question hinges on the fact that that you should know that prokaryotes, they have circular DNA. Okay, I believe it's something that we talked about before. Okay, so the DNA is a circle. Well, not necessarily, it could be an oval, whatever, but then like it loops back on itself rather than ours are like linear, right? Okay. So what happens is if you want to copy this, you're going to put maybe a primer. Oops, you're going to put a primer and then it will replicate. And when it completes the loop, it will just fall back on its own, right? So it will not get shortened like our linear DNA, which is why the prokaryotes, they actually do not need telomerase. Okay, so this is not the answer. Prokaryotes chromosomes have a single origin of replication, whereas eukaryotic chromosomes have many. That's actually correct because the prokaryotic chromosomes are much smaller, shorter compared to the eukaryotic cells. Okay, so just having one replication bubble is sufficient to perform the replication process in a relatively quick sense. You know, bacteria, they could divide, you know, under optimal conditions, they can divide every 20 minutes. Okay? But for eukaryotic cells, our chromosomes are so long, if you only have one replication bubble, it will take forever to copy the entire chromosomes. So by having multiple origin replications, you can speed up the process exponentially. Prokaryotic chromosomes has histone. We just talked about that. It's the other way around. It's a eukaryote that needs a histone. Again, because our DNA is so much longer, it will not fit if we don't package them. And Okazaki fragments is going to be found in the eukaryotes, not the prokaryotes, because once again, prokaryotes have circular DNA. Okay. I think that's all we can do for now. We'll come back to this later. Okay, let's move on to the chart. Okay, so over here we have end of interphase. Okay, so interphase is not part of mitosis. Okay, but end of interphase, we're talking about finishing G2 checkpoints already. So it's about to go into prophase. So what is going on here? Okay, so in this case, we can write DNA replication is completed. Okay. And then the chromatin starts to condense into chromosomes. Okay. And a G2 checkpoint is completed. Okay. Then we're going to go to the prophase. Okay, so a bunch of things happen in prophase. Let's write them out here. So in prophase, the chromatin is fully condensed into chromosomes. Duplicated DNA are or joins together at the centromere. Okay. The centrioles, which are these things right here. Centrioles move to the opposite pole of the cell. Nuclear envelope starts to dissolve. Okay. Oh, sorry, I should probably add something here. 1 second. Centriole moves to the opposite poles and starts to produce spindle fibers. All these lines are spindle fibers. Okay. Next, metaphase. M is for middle. Okay, so the spindle fibers attaches to the centromere of each chromosome. A tug of war between opposing spindle fibers helps line up the chromosome in the middle of the cell. Next, we have the telophase, anaphase. Okay. Spindle fibers separates the sister chromatids and pull them towards the opposite pole. And then finally, we have telophase. Telophase is backwards of prophase. So everything we wrote down in prophase, we just write them backwards here. So spindle fibers start to disappear. Chromosome uncoils. Uncoil back into chromatin. Nuclear envelope reforms. Okay. And then finally, we have cytokinesis, which is the division of cytoplasm. Okay. That's it. That's your mitosis. Okay, let's do more poll questions. Number 2 here. Let's try number 2. Please participate, okay? Choose something. Okay, I have 6 responses. Waiting for 12 more. Please choose something. Okay. Well, let's take a look at this again. Okay. How many chromosomes here in metaphase in this diagram? Oh, I told you this count as 1, right? As long as they stick together, it's called one thing. So that's 1, 2, 3, 4 chromosomes. And then in anaphase, they get pulled apart. So now we have 1, 2, 3, 4, 5, 6, 7, 8 chromosomes, right? So applying this to the question, if there are 8 chromosomes in metaphase, in anaphase, there will be 16. Okay? In metaphase, they're still stuck together like this. But as soon as you pull them apart, you count them as 2. So that's why it is going to be 16. All right. So for everyone who chose 4 or 8, have I convinced you why the answer is 16? Will you be choosing 16 on the test? I certainly hope so. Okay, number 3 is pretty straightforward. Let's do it anyways. It's one of those makes you feel good question. Like, yeah, I know the answer to this. Okay. Most of you chose telophase. That's the correct answer. Let's skip that one. Well, I will talk about it. DNA is organized and arranged in the nucleus as chromosomes, right? It's pretty straightforward. Okay, this one, let's talk about this. There's a drug called, I have trouble pronouncing this, but I think is paclitaxel, maybe. Okay, so this paclitaxel drug inhibits the formation of spindle fibers. Explain why it is used as a chemotherapy drug for cancer treatment and what might be the side effect. So cancer is basically uncontrolled. Uncontrolled cell division. Okay, it just keeps on dividing and dividing and dividing. It just keeps on undergoing mitosis. So if you have something that inhibits the formation of spindle fibers, then you will basically inhibit the cell division process, right? So you will slow down the cancer progression. The tumor is not going to grow as fast because you are suppressing cell division. But of course, unless you can deliver the drug only to the cancer cells, and we don't have the technology to do that, other cells in the body is also going to be affected. So cells that regularly divides a lot, they would be suppressed by this drug as well. So for example, cells in the hair follicle, right? They would stop dividing as much, and then people might lose hair as they undergo chemotherapy. Stem cells in the bone marrows might also be affected. So you might not be able to produce as many red cells or white cells. So you might become anemic, and your immune system is going to be compromised. Okay, so any drugs that targets the division of cancer cells would, as a side effect, also target regular cell division. Okay. Maybe I will copy these questions and let you try them on your own later, because we are running out of time. I'll put them here at the very end. Okay, you can try them later when you're bored. Okay, let's go here in the long answer questions. Does anybody have any requests for the long answer questions? If no requests, then I will do the application question. Give you a moment to let me know. Okay, well, if you change your mind, or if you thought of something else later, feel free to let me know. Email me, or just put them in the chat box if you think of it before the tutorial ends. Okay, let's take a look at this one. Answer the following questions based on the karyotype shown on the right. A karyotype is an orderly arrangement of chromosomes, right? Okay. So first question, which chromosome is the longest and which one is the shortest? Well, the longest one is chromosome number 1, and the shortest one is going to be chromosome 22. Okay, so in fact, the karyotype, how are they numbered? They're numbered based on the size, the length, right? The length of the chromosome and the position of the centromere. Okay, so you see the centromere here, here, here. So depending on where they are, right? There's the P arm and the Q arm. So there's some ratio to them, and that's how they're being, organized. Okay, so you have to be a trained geneticist to know how to organize them. Obviously, I don't know how to organize them, but I know these are the criteria that it's based on when it comes to organizing them. Is this a male or a female? What do you think? Male or female based on the karyotype? Wait a sec, male. And how do you know? How do you know it's male? Does everybody agree it is what male? Yeah, because of the Y chromosome, right? So XY is male. And then XX is female. Okay. So the father is the one who's responsible for determining the gender of the child, right? Okay. I have two daughters, and then my grandmother, who is very old-fashioned, she would ask me sometimes, like, how come your wife is not bearing any sons for you? And then I try to explain to her, it's actually my fault, okay? Because I'm the one who is supposed to be passing on the Y. I don't think she's understanding my point, but it is the male that is responsible for, well, not responsible, but like it's the male that is the determining factor, okay, of whether the child is going to be male or female. Anyways, explain whether the karyotype shows any abnormalities. Okay, any problem here that you notice? Any abnormalities here? 5? What do you mean 5? Oh, like this. No, I mean, it's just the way that it was prepared on the slide. You know, the bending doesn't really matter. Sometimes they look really awkward. Sita said number 21. What's wrong with number 21, Sita? 23, there's no 23 though, right? 23 is the 23rd pair is the sex chromosome, right? Okay. Yeah, exactly, right? So there's an extra chromosome 21, right? Okay, so this is what we call trisomy 21. Anybody know what condition is associated with trisomy 21? That's right, Down syndrome. All right, so mothers can actually undergo genetic screening. Okay, there is something called amniocentesis. Did I talk about? I don't think I talked about amniocentesis in the lecture. Okay. Does anybody here know what amniocentesis is? Any mothers here who are familiar with that process and would like to explain? Yeah, at about 21 weeks. That's the minimum.
I think I know what it is only because in my friend's family, like schizophrenia runs in her family or something. So when they found out, they had to go get like tested or whatever just to make sure like the baby would be okay. If there's any like preconceiving like illnesses or whatever, so you can like opt out, I think, or something like that.
Right. So do you know what this process does? Or anyone else is okay. What is involved in this process? No mothers here. Usually there are like a few mothers here who might be familiar with this process.
I think it's drawing amnio fluid from the uterus and then testing that.
Exactly, that is correct. Okay, so when a woman is pregnant, usually there are what we call like prenatal screening. A lot of testing is going to be done. There's blood tests, there is like ultrasound, and those blood tests and ultrasound usually would identify concerning markers, if any. Okay, so for example, if there are certain elevation in certain blood compound, that might be associated with certain risk. Okay, in the ultrasound, right, there might be some location that are like more echogenic, it creates more of a feedback on the ultrasound. That could be a marker for something else. So if there are a lot of markers, then that could be a cause for concern. And then the doctor might recommend doing an amniocentesis, which involves inserting a needle at around the belly button of the mother. Of course, it's guided by ultrasound. And that needle will take a little bit of the amniotic fluid. For those of you who don't know what amniotic fluid is, that, you know, the baby grows in the uterus, as Moira mentioned, and then there is a sac, the amniotic sac that surrounds the baby, right? And inside that sac is the amniotic fluid, which helps the baby float in the mother's womb. And that liquid contains a lot of baby's cell. Okay, so yeah, the fluid is what comes out when the water breaks. Okay, and then for those of you who don't know, when a mother goes into labor, okay, one of the things that could happen before the labor goes in is that the water breaks. Okay, so all the amniotic fluid kind of leaks out, and that's a sign that you should go to the hospital, you know, as soon as possible. Someone asked me before, how do you know the water is breaking? And then I told the person, trust me, you will definitely know. Okay. In any case, so you can take a little bit of that fluid from the needle and extract the baby's cell from it and look at the DNA of the baby. and create a karyotype. Okay, so trisomy 21 is only one of the diseases that can be screened through amniocentesis. I think there is a total of maybe 11 to 13, I forgot which is it, conditions that can be screened this way. Okay, the advantage of this is that can be done earlier in pregnancy, and so if the person decided to perform an abortion, for example, it could be done at a early on age. Obviously, that is a very like an ethical decision that has to be debated, and it's up to the person, perhaps. But this can be done earlier in pregnancy rather than later. Okay, and also the risk associated with this process is it's quite minimal. Okay, so it is something that is done routinely if the mother chooses that option. It can be corrected or abortion. Well, it's if the genetic screening come back and show that there is 3 chromosome 21, there's nothing that can be done to correct the situation, right? It's pretty much a certainty. It is a certainty, I should say, that the child will be born with Down syndrome. However, the severity of the condition might vary from individual to individual. There is no way to tell until the child starts to develop. Okay, but once the parents know about it, they could decide to continue with the pregnancy or not. Right? It's up to the woman. At least in countries where abortion is legal, right? As you know, in the States, right? For example, some states does not allow abortion to take place, and it's of course a very hotly debated topic in Congress, for example. Okay, so what's going on here? Autism also can be detected through DNA. No, autism cannot be detected through amniocentesis. But they think there is a genetic component to autism, but definitely not something that can be picked up so early in pregnancy. All right, I think that is all. I mean, I will post the fill in the blanks later so you can check it yourself. And there's just one thing left to do, which is to identify the pictures here in the diagram. Okay. So first one, we have this is a plant cell, first of all, and you can't really see the individual chromosomes yet. They are still kind of uncondensed. So I'm going to say this is interphase. Okay. Here you can see that the cell is starting to divide. It's going to cleaving into 2. So this would be telophase. Okay. T for tearing apart the cell. We have another plant cell here, and we can see that the chromosomes are very well defined at this point. We can see some spindle fibers. Okay, there are no centrioles in plant cells. Nevertheless, the spindle fibers are being formed. So this would be prophase. Chromosomes are being separated here. That is anaphase. Over here, it's again, you can see the centriole starting to move the opposite side. You get some spindle fibers. Nuclear envelope is dissolving. That is prophase. Okay, this is animal cell. This is plant cell. Anaphase. This one all lines up in the middle. Metaphase. Another metaphase. Telophase here. You can see that 2 new nuclei are forming. And this is the first real picture. This is onion cell. This is anaphase. Okay. So as far as I know, there's nothing due next week, no quiz next week. Next week we'll do lecture number 7. It is a little bit difficult. There's a lot of problem solving, especially if you have not learned it before. So please take the time to watch the lecture recordings before coming to the tutorial. And then you're going to have a quiz the week after. Okay. The lab is going to be happening later on in the month, but it's already uploaded. Okay. In fact, I have some students who already submitted their lab number 2. Okay. So if you want to get a head start on that, you are encouraged to do so. Okay. You don't have to hand it in per se, but you should, at least start on it. Adriana asked, yes, the next quiz will be on lecture 6 and 7. Okay, 6 and 7. Okay, well, thank you very much for coming. And I hope you guys have a good weekend. And I'll see you again next week. Bye for now.